Welcome you all students. I think all of you are doing fine. So from this video onwards, I'll be discussing about quantitative genetics. So what do you mean by quantitative genetics? Quantitative genetics usually deals with very important economical agronomic characters. Got it? Which are inherited by multiple genes, hence they are complex as their analysis is slightly difficult comparing to the monogenic characters. So these characters are estimated in quantities, hence whichever the character which can be estimated in matrix or metrically are called as quantitative traits, and the study which deals with analyzing these traits are called as quantitative genetics. Is it clear? These quantitative genetics includes the study of height of an organism, weight of an organism, yield of a particular organism with respect to either the number of seeds produced or the type of seeds produced. Example, the milk production in case of cattle. Okay, so you can take many examples of these types which are economically very important, and they have to be estimated for breeding purpose. Is that clear? Okay, next, what are the characteristic features of this quantitative traits? These quantitative traits show one specific character, that is continuous variation. What do you mean by continuous variation? To know that, we have to know what is discontinuous variation. So in discontinuous variation, you can see very clearly there are two phenotypes. One is tall, the other one is dwarf. So what type of character it is? It's a Mendelian character, where you will see only two types of phenotypes. One is dominant phenotype, another one is recessive phenotypes, and the number of dominant phenotypes will be always more than the number of recessive phenotypes. Is it clear? And these characters will continuously inherit in the same manner without showing any changes in between. The characters remains constant generation after generation without undergoing any change. This is the feature of discontinuous traits. What is the characteristic feature of a continuous trait then, which are called as quantitative traits? So these characters vary continuously and are controlled by multiple alleles. Vary continuously and are controlled by multiple alleles. Estimating their genotypic and phenotypic relationship. Remember, estimating their genotypic and phenotypic relationship for anybody is very complicated. Hence, we also refer them as complex characters because we cannot discrete them or segregate them into specific categories like the tall or the dwarf. Is that clear? They do not just give difficulty with respect to analysis of genotype or phenotype, they also involve the environment in them. So their influence, the influence of environment on this multiple alleles will be very high. So what they do, they express with a cumulative range, which we call it as cumulative effect. That is the combination of genotype, environment, gives rise to a phenotype, which is a cumulative effect of the two. Got it? So when a character is expressed in such a way, there will be many number of variants formed between the dominant and the recessive. Is that clear? So all the characters can be put into a binomial distribution from the tallest to the shortest. In between that, there will be range of variants. Is that clear? So this is the characteristic feature of a quantitative trait. Next, types of quantitative traits. So usually quantitative traits can be divided into three types. One which shows continuous variability irrespective of the matrix dynamics. So what does it mean? That if there is a quantitative character, the quantitative character can range in any way. Example, if you take the amount of milk produced by a cow, different cows can be 1.5 liters, 2 liters, 2.5 liters, 5 liters or 6.5 liters. Like that, there is no specification that it has to produce only 1 liter or 2 liter like that. Is that clear? So any matrix can be applied into these type of characters. That is called as, they are called as normal quantitative traits. But there are few traits which shows continuous variations, but all matrix dimensions cannot be applied on these traits. They can be only described with whole numbers. They do not have an infinite range of phenotypes, like they will not have 1.5, 2.5, okay, with respect to the yield, or the number of leaves, okay. The number of leaves in a stem can be 5 or 6 or 10. It cannot be 1.5, 2.5 or 3.5 like that, okay. Got it? So that is the uniqueness of this quantitative trait. These type of traits are called as meristic traits, not metric traits. These are called as meristic traits. Got it? Example, seeds in a pond, number of seeds in a pond, number of eggs laid by the chick, okay, or chicken, or the number of bristles present in Drosophila can be 5, 10, 15 like that. Not more, not 5.5, 10.5 or 15.5. Is that clear? So this is one type of quantitative trait. The next type of quantitative trait, which is also continuous in nature and shows only two types of phenotypes. One is either the character will be expressed or character will not be expressed. There are only two types, like that of Mendelian traits. But in spite of that, we classify them in quantitative trait because we are unable to specify a particular gene for expression of this trait, and they are medically very, very important. Example, blood pressure, okay, immune disorders in case of human beings, diabetes mellitus, type 2 diabetes mellitus. So these are the characters. These characters are polygenic in nature and multifactorial. Polygenic, many number of genes are involved and many factors are involved in these expression of these characters. Got it? And when these characters starts to express, only when the genotype reaches a threshold level and is influenced by multiple factors. When a particular individual reaches a threshold and results with the effect of multiple factors. So what are those multiple factors? It will be environmental factors. Got it? It can be food, exercise, diet, whatever, lifestyle, many numbers. Got it? So what happens? We can divide the population into two discrete categories. One with a healthy population, another one with a diseased population. Is that clear? In the diseased population also, there will be range of phenotypes with the limited expression to a complex expression, severe expression. Is that clear? So this is the speciality of this threshold quantitative trait. Is that clear? So there are two types. One is meristic, another one is threshold. Is that clear? So what you can see, example, you can see greater or lesser likelihood of developing the disease when multiple genes and multi-factors are involved in expressing a particular trait. Okay, so let's analyze this with an example. So as we said, there is a genotype which will have a threshold capacity. If it exceeds that genotype will interact with the environment, okay, and that results in expressing the character. Example, tuberculosis type 2 diabetes mellitus. So how tuberculosis will result in creating two types of populations, one with the tuberculosis, another one without tuberculosis. Because the individuals who get tuberculosis are, most of them, if we put them into categorize them, they are poor, who are exposed to disease again and again, and who do not complete the doses given to the individuals, okay, who are already been affected with. So they will become the spreaders. So whoever is around them, the probability of getting the disease will also be high. The same way, the individuals who have a tendency of genes to get diabetes, that becomes fastened when the threshold level is reached, and multiple factors will start to affect. That is the diet, lifestyle, everything. Is that clear? So how to improve this? Yes, there is a possibility we can improve by knowing the genotypes well, and the physical environment influencing this genotypes can help in reducing the expression of these diseases. Is that clear? Okay, next, let's see. So after knowing about the polygenic trait and the quantitative traits and we have to differentiate between quantitative traits and qualitative traits. So let's see what are the differences we can come up with. One is single gene inheritance or oligogenic or Mendelian or monogenic traits are discontinuous in nature. They do not show much variations, so hence they show discontinuous variation, and one gene is more than enough to form the complete trait. Either they will be dominant or recessive. In case of polygenic, they produce continuous variations, and the number of traits produced is many in number. Got it? So the number of traits produced expressed is many. That a dominant allele controls the complete expression of a trait, of a qualitative trait, whereas here dominant allele is just a unit of the total expression. It's just a unit of total expression. So number of dominant allele, there should be more number of dominant allele to produce more dominant effect. One single allele cannot determine the effect. Is that clear? So here recessiveness also has equal effect. Then a character is presented in an individual by pair of alleles in diploids. Okay, either dominant or recessive, or they can be in the form of heterozygotes. Here it's not like that. Multiple genes are present in multiple loci. They all will group up, will produce enzymes or factors which all put together will result in expressing a character or repressing a character. Is that clear? So multiple alleles are involved in expressing a character. F1 individuals are similar to dominant parental type. So if you take two Mendelian characters and cross, the F1 will be the dominant type. But here it's not like that. The F1 will be an intermediate type and not a dominant type. Is that clear? And that's the same in case of no intermediates are produced. Here intermediates are very, very common, and there will be range of intermediates. The range of intermediates. There is no cumulative action. Got it? No dominant will mix with recessive and expresses a trait. Here there is cumulative action. There is a cumulative effect. So there will be range of phenotypes produced. Okay. So individuals, in case of qualitative traits, dominant phenotypes are more in number, always in the ratio of 3:1. Here there is no dominant or recessiveness. Instead the range will be, I will be discussing later on, that is the ratio will be 1:4:6:4:1. Okay, so there will be, you can see the range in between. 1 is dominant, 4 is again intermediate, 6 is further an intermediate, 4 again a different type of intermediate, and 1 is one more type of intermediate. So 1 is to 4 is to 6 is to 4 is to 1, and the last one is also is recessive. Is that clear? So like that, range of phenotypes will be produced in case of qualitative traits. Okay. So with this, I think you people would have understood exactly about the quantitative traits. Okay. So you should be able to, after this video, you should be able to define a quantitative. What is quantitative genetics? What is a quantitative character? What are the characteristics features of a quantitative trait? What are the characteristic features of a qualitative trait? Mention the types of quantitative traits. Define a threshold trait with an example. Differentiate between polygenic and monogenic trait. Is that clear? Got it? So with this video, you should be able to answer all these questions. Thank you all.